Today I'll be presenting on the development and reliability of a quantitative personal hygiene assessment tool, or qPHAT as we called it, and implications for holistic WASH program evaluation. I think this goes without saying in a forum such as this, but personal hygiene practices, including face washing and hand washing, are important for preventing exposure to and transmission of various infectious diseases. But the issue here is that they're often very difficult to measure. Many cleanliness measures actually reflect proxy indicators of personal hygiene behavior. Conventional qualitative metrics tend to yield binary outcomes that may lack the nuance necessary for actually distinguishing between various levels of cleanliness. And because behavior change is often incremental, we should really be striving for metrics that are sensitive to incremental change so we can actually monitor and evaluate that change over time. And while it's important to develop reliable metrics, it's also important to note that reliability does not equate to validity or utility. Cleanliness metrics should generate valid measures of cleanliness and also predict recent cleansing practices. So that's something important to consider as we move on in the presentation. Okay. Work I'll be presenting today reflects a sub-study that was embedded within a larger cluster randomized trial called the Delea trial. The purpose of the Delea trial was to evaluate the impact of a demand-side sanitation and hygiene intervention on sustained behavior change and mental well-being. This was conducted in Amhara, Ethiopia. And as a result, because that was our purpose of the study, we were particularly interested in ensuring high-quality measures of the uptake of improved sanitation and hygiene practices. And we wanted to potentially improve upon existing metrics used to assess facial and hand cleanliness. The trial was conducted in 50 randomly selected sub-districts in 3 districts within West Gojam and South Gondar in the Amhara region of Ethiopia. And we randomly selected approximately 30 households with at least one child aged 1 to 9 years per study cluster. And this child was indicated as the index child. To enable better assessment of personal hygiene practices and enhance evaluations of personal hygiene interventions, we sought to create a metric that generates quantitative data on facial and hand cleanliness. The aim of this study was to develop and pilot a novel quantitative metric to assess the reliability of that metric and to also compare the reliability estimates and measurement attributes of the conventional qualitative facial and hand cleanliness metrics against this novel metric. And we specifically sought to examine whether the novel metric was reliable for measuring facial and hand cleanliness, and to determine whether the reliability estimates differed meaningfully across different types of readers, including those who were not blinded to the conditions of the household and the child, so the people who were actually collecting the data, as well as people who were blinded to those conditions, because we thought that there might be a bit of bias introduced as people were actually seeing the conditions and of the household and seeing the conditions of the child when they were assessing the cleanliness of that child. And we were also interested in understanding how human generated rating scores compared to computer simulated rating scores. In order to investigate our research questions, we needed to develop a metric that would allow for quantitative assessments of facial and hand cleanliness. So we used color theory principles to create a novel standardized color scale to do that. And this scale depicted a continuum of color along the RGB, or red, green, blue color model array. And this ranged from 10 being the lightest color along the continuum to 0 being the darkest color along the continuum. And each point along the continuum represented a 10% increase in the saturation of the color along that RGB color model array. This yielded an 11-point qPHAT color scale that we used. And after conducting some formative research to test and refine the color scale to basically understand what color model array we needed to define, we used the qPHAT methodology to obtain quantitative proxy measures of personal hygiene through facial and hand cleanliness assessments. In addition to using this color scale, we also used sterile gauze pads that were pre-moistened with saline solution to collect wipes of hands and faces. We also developed and tested a standardized process for collecting those wipes of the face, so the area around the eyes, and the hands of one index child aged 1 to 9 years per household. And then raters were trained to use a standardized process to rate those wipes against the qPHAT color scale. So they collected the wipe from the child's faces and hands by tracing the face and hands. And then about an hour later, so at the end of a household survey they were conducting, they scanned the wipe for the darkest square half inch of the wipe. And then they scored that darkest area against the qPHAT color scale to identify the most similar color within that scale. And I'll explain in a little bit more detail in subsequent slides, but we also used a similar method to identify and score the darkest area of the wipe against the qPHAT color scale when we conducted our densitometric assessments, which was the computer simulated rating scores. Right. So we designed our study to examine inter-rater reliability of rating scores across not only different rating groups, but also different cleanliness measures. So enumerators collected data from one index child per study household, and they collected those data. They started out with obtaining conventional qualitative measures of facial cleanliness. And this was measured by the presence or absence of ocular discharge, wet and or dry nasal discharge, dirt and debris on face, and the number of fly face contacts during a 1-minute observation period. These are standard qualitative metrics that are used in the trachoma community for assessing facial cleanliness. And for hand cleanliness, we looked at the qualitative measures as measured by presence or absence of dirt under nails or on palms and or finger pads, with each hand assessed separately. And again, this is a typical qualitative hand cleanliness metric that might be used by some wash professionals. After collecting data using the conventional cleanliness metrics, enumerators also collected wipes of the index children's faces and hands, and they used the qPHAT color scale to generate the quantitative ratings of facial and hand cleanliness. In addition to those enumerator ratings, field supervisors also simultaneously yet independently use the conventional qualitative metrics to generate data on facial and hand cleanliness from a validation sample of approximately 10% of households per study cluster. That allowed us to assess inter-rater reliability between non-blinded raters who were collecting the data in the field. Because we were interested in understanding whether ratings generated by individuals who were not blinded to the conditions of the household and the child were different from individuals who were blinded to those conditions, we had a master blinded rater use the qPHAT methodology to rate facial and hand wipes in an off-site setting. So this was at a regional office. We also had 2 additional raters who were blinded to the conditions of the household and child rate a subset of those wipes to examine inter-rater reliability amongst blinded raters. And finally, Jed Snyder, a study team member, had the brilliant idea of using densitometry to generate computer simulated rating scores of a subset of those wipes that were assessed by the blinded raters. And I'll talk in a little bit more detail about densitometry in a minute. For our analyses, we generated descriptive statistics on conventional and qPHAT cleanliness data. For the conventional qualitative metrics, we assess prevalence of individual signs of clean face and clean hands, while also assessing a composite indicator of clean face or clean hands. For the qPHAT rating scores, we assess the distribution of scores across an 11-point qPHAT scale. And because kappa statistics are often criticized, we use Gwet's agreement coefficient to generate chance-corrected agreement coefficients that adjust for the number of rating categories and the frequency with which they are used by raters. We also use quadratic weights to assess partial agreement between in the qPHAT rating scores, and we use probabilistic benchmarking and the Landis and Koch benchmarking scale to interpret our inter-rater reliability results. As previously mentioned, we used densitometry to generate computer simulated rating scores so that we could compare those scores to human generated rating scores. For those of you who might not be as familiar with densitometry, it's a method that is commonly used in bench lab science to assess optical density. And we actually adapted an open access ImageJ protocol for the purposes of our study. And we normalized lighting between images by the use of a clean wipe, which was used as a negative control for calibration. Then the wipes were scanned for the darkest square half inch within the wipe. And this was a similar approach that the human raters used when they were in the field, rating the wipes. And then through the use of densitometry, histogram values of the qPHAT color scale were compared to histogram values of the wipe images using the 5th percentile of the sampling frame. So this kind of simulated the scoring of that darkest area of the wipe that mirrored the human rater protocol. And here's just an illustrative sample of the wipes that we collected. And as you can see, the density of the color and the cleanliness of children's faces varied across our sample. For my results, in the interest of time, I'll just be focusing my discussion today on results related to the inter-rater reliability of the qPHAT metric. So this slide here demonstrates that for facial and hand cleanliness, enumerators and master blinded rater had very similar rating scores that were highly reliable. And so you can see that if you look at this third column, IRR estimate, the inter-rater reliability coefficients are demonstrated here. And these range from 0.9 for facial cleanliness to 0.92 for hand cleanliness. Where an IRR estimate of 1.0 would reflect perfect agreement. So we can see these are quite high. In this next slide, we see that the 3 blinded raters also had similar rating scores and high inter-rater reliability. If you look at the 4th column here, you'll see those coefficients. And they range from 0.95 for hand cleanliness measures to 0.96 for face cleanliness measures. And the inter-rater reliability of the computer simulated and blinded human generated facial cleanliness rating scores range slightly by rater. But if you look at this 5th column here, you can see that they range from 0.96 to 0.98. And these were consistently high across all blinded raters. Finally, we take a look at hand cleanliness, the inter-rater reliability of computer simulated and blinded human generated hand cleanliness rating scores. And these ranged from 0.96 to 0.97 for the reliability, inter-rater reliability, which was also quite high. So in summary, our research group developed a novel cleanliness metric that allows for quantitative measurement of facial and hand cleanliness. The data generated by the qPHAT methodology actually produced data that are finer in granularity than conventional qualitative cleanliness data. And having these more granular data could potentially facilitate better monitoring and evaluation of the uptake of personal hygiene interventions, and that are not typically afforded by coarser conventional qualitative cleanliness metrics. So in other words, these proxy indicators of personal hygiene could improve upon the quality of data available for conducting program evaluations and assessing intervention effectiveness. With regard to next steps, prior to COVID-19, we were in the process of launching a multi-country study to assess the reliability of the qPHAT methodology in context within and outside of Ethiopia. We were examining the validity of the data generated by the methodology. So for this initial study that I just presented, we were focused on looking at inter-rater reliability. It was outside of the scope of our study to actually also examine validity of those measures, but we are in the process of launching a study to do that, where we will look to see that this methodology ensures that measures are indeed valid indicators of recent cleansing practices, which some of the conventional qualitative metrics, particularly those related to facial cleanliness, actually in studies have shown to not be valid indicators of recent practices. So we'll be investigating that a bit more. And we'll also be exploring the utility of different types of hygiene measures and investigating whether cleanliness, as measured by the qPHAT methodology, reflects risk factors for diseases such as trachoma. And that's it. I'd just like to acknowledge and make note of the larger team of individuals involved in this research, as well as our funding agencies.